I would like to acknowledge all the people who make my life very interesting. People who help me in my research, and all of them been involved in gamma-radiated vaccines, starting with Arno, formerly at ANU, now James Payton at Adelaide, and my excellent first PhD students, Dr. Yuichi Furuya, is now in America, and current PhD students, Rachel Bab, and also Professor Tim Hurst, who helped establishing gamma vaccine, which has been involved in supporting my research. So, if I ask the audience a simple question, if I give you this vaccine and say this being gamma radiated, so who will not accept to take this vaccine? Do you have any reservation? If no one will reject this vaccine, then my life is easy. Because people's perception for gamma radiation is crucial to accept those vaccines. And because you're all scientists, you did not object, and to really tell you this is completely sterile and safe vaccine, my life is easy, then we need to talk to the public to see their perception. So, I was thinking to talk about concepts of nuclear physics and how we could apply those concepts to develop vaccines. But yesterday, Alan talked about a new mucosal vaccine that induces gamma delta T cells, so it's not even adaptive immunity. And I thought I should really bring that on board, because that work, which you saw last week in Oxford, it's a work of Rachel Bab, who's my PhD student, so I thought I should bring the excitement. Alan was excited yesterday to talk about it, and I thought I should bring pneumococcal vaccine as well. So to start with gamma radiation, very safe procedure, being examined thoroughly by WHO, by FAO, European, the Americans, and also in Australia and New Zealand. We have approval to use gamma radiation to sterilize tropical fruit. Definitely mangoes, kiwis, any kiwis imported from New Zealand into Australia exposed to gamma radiation. Any mangoes from tropical islands will be exposed to gamma radiation. Persimmons as well. So it's a very common strategy to sterilize food. There are two important concepts here. The first one, to inactivate bacteria and viruses, so those foods will be safe and sterile. The other one, to sterilize insect pests, so basically prolong the shelf life of food. We all have been consuming irradiated food in Australia. Put it this way, if you go to Woolworth or Coles and buy dates, that dates will be exposed to gamma radiation, 50 kilograms. So we all been consuming tons probably of irradiated foods. We're all okay. So that is the first one. Many years ago, I started working on gamma radiated melanoma vaccine, and at Monash, and I told my supervisor that if I want to develop gamma radiated melanoma vaccine, we should make it as therapeutic. We cannot make a cancer vaccine as preventative. If I want to work on preventative gamma-radiated vaccines, I should work on viruses. That's why I moved to ANU and started working on gamma-radiated vaccines. What's interesting here is there's a clinical trials for gamma-radiated viruses. This published 4 months ago, in March, I think. They talk about using BCG vaccine, gamma-radiated BCG, and that clinical trials was looking for sepsis-induced immunoparalysis, and they want to restore the immune response in sepsis patients. So they did use gamma radiation because they could not use live bacteria. They already immune paralysis. There's immune paralysis in those patients. So what's interesting here, there's a 90% immune response to gamma-radiated BCG. This is the key message here. That's the clinical trials in human, and the vaccine injected intradermal. The other one is gamma-radiated melanoma vaccine. This is the first vaccine I was working on. It's interesting to see. It's a new version of that vaccine coming into clinical trials. I was working on it in Monash. What they did, they administered this vaccine intradermally, and what they found is it's safe and tolerable, so people could tolerate those vaccine. And that vaccine was for people who went under surgery for melanoma, so it's more therapeutic prophylactic vaccines. So these clearly tell us that gamma-radiated vaccine is coming. And I thought people's perception for gamma radiation activated vaccines, if we understand the benefit of it, I think we'll take a way forward and we'll see more vaccines. So the sterilizing effect of, this is very crucial for us to understand the sterilizing effect of gamma radiation. Gamma ray is, if we look at the electromagnetic spectrum, it's the high end, so it's a high energy, short wavelength, destroy the genetic materials, creating nicks in the genetic materials, so basically render those genomes inactive, so because you destroy those genes. There is also indirect effect. So the first one is the direct effect when we hit with the photons, we hit the genome and destroy the genome. But there is an indirect effect, and everybody was, the reluctance to take gamma radiation forward many years ago is the indirect effect and the free radicals, which I'm not going to talk about. But what's crucial here, we could control the indirect effect by controlling the condition of radiation. So basically, if we use low temperature during the radiation process, we could eliminate or reduce the indirect effect. So by reducing the indirect effect, we maintain protein integrity and pathogen integrity, and that's crucial to maintain the integrity of those pathogens to induce proper immune response. So gamma radiation has been used to sterilize many highly pathogenic, highly infectious agents, such as Ebola, Lassa, chikungunya in Australia, H5N1. My colleague, former colleague at SABAth, was working on gamma-radiated H5N1 from CSIRO and was developing a reagent for ELISA. And I told him, this is my universal vaccine. I've been working on gamma-radiated vaccine for many years, and now you're using H5N1 gamma-radiated virus as an antigen. So it has been used to inactivate highly infectious agents. So I will talk about flu first. This is just to remind you, so I'm not going into great detail, but this is just to put the story together. So flu, segmented genomes, there's internal proteins, mainly the nuclear protein and matrix. Most of the vaccine target in terms of antibody response targets the external part of the virus, which is HA and NA. But what is inside is the matrix and the nuclear protein. These are the target for our T-cell responses. They mainly conserve, and all of us will respond in terms of CD8 T-cell responses, either to MP protein, the nuclear protein, or to matrix. So we will have a good CD8 T-cell response against those internal conserved protein. If we think about the adaptive immune response to flu, the first arm is the antibody responses, which mainly target the external part. The cell-mediated immunity target the virus-infected cells and need to have MHC class 1 presentation. And that small peptides, about 9 amino acids, need to be derived, may be derived in flu from the internal part. So the CD8 T cell, therefore, is more cross-reactive because the antibody response target the external part, and that is affected by mutation, while the internal part is more conserved. And that, therefore, if we induce CD8 T cell responses, we're talking about cross-reactive vaccine. And current vaccine, we're talking about inactivated, so I'm not talking about live attenuated vaccine. Here I'm talking about inactivated, so not the live attenuated. So 3 different vaccines. The first one is the formalin inactivated, because it's formalin, the virus becomes very rigid, can only induce antibody responses. There is no CTL responses or cytotoxic T-cell responses. The syphilis vaccine, which we use in Australia, it's basically been destroyed with detergent, beta-proviolactone and ether. And because it's syphilis, we don't have the structure that allows MHC class 1 presentation. So this is antibody response only. In America and Europe now, they use the subunit vaccine, which also induces antibody responses. So flu mutate frequently, and that's why we have seasonal and pandemic. And the flu vaccine, the WHO recommend changes to those flu vaccine very frequently. So if we think about our current vaccine and WHO structure to recommend for vaccination, We change those strains very often, and the system cannot predict the upcoming pandemic. So yesterday, we heard from Nick about the vaccine for H7N9, but that's what we know that causes the species barrier. In 2009, no one could have predicted the H1N1 that caused species barrier and caused pandemic, because the system is not designed to predict the new pandemic strains. Yes, we could detect it when it's already crossed the species barrier. Unless we go to the wild and we start to really screen what's around in birds. And in birds, there's about 144 different subtypes. With each subtype, there will be an enormous number of mutations. So within the subtypes, there will be a limited number of strains and mutations. So the system is only designed to chase those mutations in human. So what really happening in circulation, so we have to adapt our system for the upcoming season. What we need a vaccine that reacts against the flu regardless of the mutation, regardless of what type of virus could cause the species barrier and cause pandemic, whether it's H5N1 or H7N9 or different viruses. We need a vaccine that is cross-reactive. So if we think about flu vaccination, current one, there's either inactivated, that is basically inducing antibody responses, or live virus. There's a limitation for live virus, which I'm not going into, but everyone is calling for a universal flu vaccine. Can we get a universal flu vaccine? From my own understanding, this will be very limited. What we're looking for is a cross-reactive CD8 T-cell based vaccine that give us a window to survive in case of a pandemic. Give us a window so our immune system could clear it until we have good antibody response to clear the infection. This is basically what we're talking about. And that would be our best hope. Gamma-radiated flu vaccine is meeting those requirements. We've done enough work on it. I'm very confident this will be in clinical trials soon, probably in the next few years. Soon in terms of vaccine development means a few years. So let's think about the first one. The first study we did with gamma-radiated vaccine, we have flu virus. We grow it in eggs. We concentrated using chicken red blood cell. This is, of course, you cannot use it for a human, but that's what I did in my lab. And then exposed to gamma radiation. We test sterility of those preparation, and I administered the vaccine using different routes. So, and then I challenged with a completely different virus. So the vaccine base is H3N2. The challenge strain is H1N1. And what we see, so this is the unvaccinated mice. We're looking for weight loss, and they were sacrificed when they reached about 30% body weight loss. And if we give IV administration, most of them die. Some of them survive, but very late. And if we give intranasal vaccination, all mice survive, losing a little bit of, sometimes they lose some weight, but they recover. We also test different routes of administration. What we found here is that intranasal administration very effective in inducing cross protection. That's basically the message here. We published many papers, so I'm just picking a few key data to present to you. So the first one here is done by Yuichi, for example. We looked for homotypic or homologous protection. And here we compare different routes, different inactivation methods. So can we use formalin as a mean to inactivate the virus? And we compare it to gamma radiation. So formalin, UV inactivation versus gamma radiation. And also we looked at the current vaccine in terms of inducing good protection after intranasal administration without adjuvant. This is basically what we're talking about. So what we found here for homotypic, if we compare gamma-radiated vaccine to UV or formalin, formalin and UV did not really induce any protection. You could see mice after a single dose and then a challenge, they're losing weight and they did not really survive. But gamma-radiated vaccine provide very good homotypic protection. So this is meeting the requirement for seasonal vaccine. Then as I showed you before for heterotypic challenge, we also compared formalin and UV inactivated vaccine compared to gamma-radiated vaccine. And what we use H1N1 as a vaccine, and a challenge strain is, Yes, the vaccine is, I should say here, is H3N2, sorry. No, yes, all right. So the vaccine is H3N2, and the challenge is H1N1. So it's reverse to the one I showed you earlier. And what we found is only the gamma-radiated vaccine inducing cross-protection after a single challenge. So this is basically, you could use H1N1, you get protection against H3N2, or you could use H3N2, you get protection against H1N1. But the real challenge is to take it to the pandemic strains, to take this vaccine against H5N1, H7N9, or any other newly emerging viruses. And that work has not been done at CSIRO, and we have a vaccine based on H1N1, and we're challenged with H5N1. And what we could see here, that those mice that have not been vaccinated, when they challenge with H5N1, they lose weight and they die. But those ones which being vaccinated with gamma-radiated H1N1, they survive H5N1 challenge. So this was very exciting data at that time when we got those data, because basically we are using a whole killed vaccine, giving that vaccine intranasally without adjuvant, and we have protection against pandemic strains. So this mechanism of protection, we published many data, and we know it's a CD8 T-cell based vaccine. And I'll show you some data, because if it's a CD8 T-cell vaccine, then we think it's to be not a sterile immunity, it's rather than a quick recovery. And that is true. If we look at the virus cycle in the lung, and for the non-vaccinated mice, we've done it many times, even for H5N1 challenge, but this one I'm showing you the H1N1 data. You could see we could detect virus in the lung by day 3 and by day 5. But those mice that have been vaccinated with H3N2, gamma-radiated H3N2, we could detect some virus in the lung by day 3, but by day 5, there is no virus in the lung. So very quick recovery, very quick clearance of the virus in the lung. And that clearance is mainly mediated by CD8 T cells. So if we did adoptive transfer for B cells and T cells. So we vaccinated mice, we got B cells and T cells from vaccinated mice, adoptively transfer those B or T cells into a new mice and challenge them. And what we see, if you transfer B cells, there is no protection. If you transfer T cells, there is a protection. So clearly the cross protection is mediated by T cells. The difference here is we give T cell in IV, and when you challenge them, there will be a time for them to be recruited into the lung to clear the infection. That's why you see a difference in the kinetics. But that's the clear data to tell us this is a CEA-based protection. And also we looked at the T cell responses in the lung. So if we don't have vaccine, what we could see by day 7, we could detect some T cell responses in the lung. This is basically the proper kinetic for T cell response in the lung. When you are infected, the antigen-presenting cell will take the antigen, go into the draining lymph node, activate T cell responses, then they will take time to go back into the site of infection and start clearing the virus-infected cells. So normally it takes about 7 days for those T cells to appear in the lung, in mice. So if we vaccinate, we could see that response very early. We can start to see it by day 3 and by day 5 clearly there. That's really match early clearance of virus-infected cells. I just showed you earlier. So if we think about CD8 T cells and providing protection and CD8 T cell response induced by gamma-radiated vaccines, the concept here is a vaccine acting like a live virus and delivering antigen into MHC class 1 presentation pathway. This is the key. So the virus acting like a live virus, binding to the cell, deliver the internal part of the virus into the cytosol and inducing antigen presentation through MHC class 1. We confirmed that one. We published data to say that the flu, gamma flu, activate the cytosolic receptors, which means there is interaction between the inactivated genomes with the cytosolic receptor to induce interferon type 1. With that one also, we illustrated that if we take RMA cells, we could sensitize those cells by gamma-radiated vaccine, but not by formalin or UV, to be a good target for T cell responses. And if we have RMAS targets, this is a mutant in TAB, there's a mutant in TAB, there will be no MHC class 1 presentation, that sensitization disappears. So clearly, those gamma-radiated virus delivering antigen into MHC class 1 presentation, and that is key to induce T cell responses. So we also looked at the structure of gamma-radiated virus. So this is exposed to 25. We have also many pictures for 50, 35, but this is a good data done by my PhD student Shannon. And what you can see, this is the H1N1, the A California that caused pandemic. This is the picture, and that is a gamma-radiated H1N1. Clearly, there's no difference. So after radiation, the integrity of the virus maintained, allowing antigen delivery into MSC class 1. So we are excited about this. I think the approach could be applied to other vaccines. What's interesting here, Nick was talking about inulin. I think it's a great adjuvant. Gamma flu, we don't need adjuvant working on its own. But what's good about gamma flu can act as an adjuvant in its own right. So we published that work. This is about the tissue resistance T cells. Before getting to the adjuvant part, there are a few. When we started working on flu, we thought about recruitment. At that time, we were dissecting why intranasal is much more effective than IV, and 2 papers published talking about tissue residence T cell in the lung after activation, and that's why you have very early response. And as I mentioned, we work gamma flu acting as an adjuvant in its own right, and we published that work. We use gamma-radiated symblyphosis virus as an antigen. This is good antigen in its own right, does not induce interferon type 1 compared to flu, which is surprising, talking about single-stranded RNA virus. When you inactivate it, you destroy the ability to induce interferon type 1, but that is a unique feature to SFV. When we mix gamma SFV with gamma flu, what we found is enhancement in antibody responses to gamma SFV. So gamma flu acted as an adjuvant to a co-administered vaccine. When we reported those data, and we had the idea, if we talk about gamma flu, we talk about respiratory infection. So the best model will be looking at other respiratory pathogen, and that's a collaboration with James Payton. So we said if we have a good universal flu vaccine, will definitely impact on the pneumococcal infection. Because pneumococcal, there's a synergy between viral infection and secondary bacterial infection, because the primary virus infection will affect colonization, so promote colonization, because it affects cellular function and epithelial destruction, increase colonization, and also impact on clearance of pathogen. And that really, there is a huge literature about how primary flu infection or virus infection promote secondary bacterial infection. But if we think about a universal flu vaccine, we need to think about secondary bacterial infection as well. If we think about gamma-radiated flu, there might be a possibility to develop gamma-radiated bacterial vaccine. And that's the collaboration with James Payton. And the candidate is Streptococcus pneumonia because it's the most common in secondary infection. In fact, in 1918, the pandemic, the 50 million deaths at that time, about 100 years ago, and mainly related to secondary bacterial infection. Streptococcus pneumonia is a major one. So we thought this would be a great candidate. We had talked yesterday about many mucosal and how Streptococcus pneumonia could be involved in that one. And when we talk about Streptococcus pneumonia, we need to think about pneumonia or bacteremia or meningitis. So the epidemiology of Streptococcus pneumonia is mainly developing countries, and probably developing countries, as Nick was saying, is they need to think about their own way to get their vaccine to protect their own people, because probably in Australia we don't have that major issue compared to those countries. So currently there are 2 vaccines. There's a problem with pneumococcal vaccine. There are 93 different serotypes, so it's a similar scenario to flu, when we have a limited number of viruses that could change. Here we have 93 different serotypes. The 2 vaccines, the polyvalent one that has 23 different serotypes, and the Prevnar has 13 different serotypes. Those serotypes, they induce serotype-specific immune response. Those vaccines don't induce cross-reactive immune response. So the problem with those vaccines If they are effective in preventing colonization, then you have serotype replacement. And current epidemiological data indicate that if you eliminate those available, two more minutes? Okay. 2 more minutes. Let's see. Okay. So the problem with current vaccine is if you eliminate the current colonization, then you have a serotype replacement. So you're not eliminating, and you need to change the Prevnar again. And also, these vaccines are not very effective in young children. So there is a high cost as well. We develop genetically modified bacteria, so I'm not going into that one, but we have very safe one. We're even working on it now. Shannon tried to introduce more mutation, but this is very effective strain. We did gamma radiation, and what you could see that integrity of the gamma radiated pneumococcal vaccine. So basically the same structure as the non-irradiated bacteria. So that's one. And then we looked for intranasal vaccination. We tested against bacteremia. So this is a homotypic challenge. So XTL1, the vaccine strain, is the same as D39, and what we could see, significant increase in survival. We also tested against completely different serotype, and we could also detect heterotypic protection, so increased percentage in terms of survival. So that's a complete different challenge, and we could see significant protection. This is the work done by Rachel, I don't know what I was talking about. And we also looked for colonization, whether the vaccine prevents colonization. And what's interesting, we have different models for this type of infection. So we found the vaccine is not really having any impact on colonization, but really preventing pneumonia. So the vaccine we have could protect against pneumonia, could protect against bacteremia, and as well as meningitis, but will not affect colonization, which is, we think it's important to really have a license for this vaccine. That's what the FDA wants. Gates want everyone to prevent colonization. FDA want to really doesn't care about colonization. We want to protect against the disease, and that's what the vaccine is doing. And the data Alan was talking about, this vaccine working by inducing gamma delta T cells, IL-17 dependent gamma delta T cells, preventing neutrophilia. There's no inflammation, but there's a clear increase in gamma delta T cells. That's it. Thank you.
So, Mohammed, you mentioned with the gamma flu the dose of HA. Are these mice getting 2 doses intranasally or 1 dose?
With this vaccine was 1 dose with gamma radiated flu, so it's 1 dose, no adjuvant, and the dose is low. I think we currently using about, we have about, just to remember, because we've done many different experiments, so it's about, I think, 1 microgram per mouse, and we can sometimes even lower.
Okay, so
with a homotypic you need lower, but with a complete heterotypic you need a bit more to induce T cell response.
So the follow-on question is that this is a whole cell vaccine, and CSL have had to withdraw their flu vaccine for children in Australia because it was incompletely split, and as a consequence was causing febrile convulsions in the children. This is also a whole cell vaccine, probably more whole cell than CSL. Isn't that going to be a fundamental problem? You're not going to be able to give this to children.
No, I think you need to think about it differently. CSL vaccine giving intramuscular injection, while this vaccine will be given intranasally. And we are exposed to many pathogens in our nose, and the vaccine will act like a live virus, so you don't have that exposure to B cells or reactogenicity compared to other, even children will be exposed to live virus, and the virus could cause some sickness. And live attenuated vaccine, for example, inducing limited sickness. So we cannot really talk about, or you have the right to question whether this vaccine will be effective.
Have you tested it for pyrexia? So if you give this vaccine intramuscularly or intranasally in mice, and you measure their temperature,
No, we did.
have you actually done that? Because I think it's a really important thing.
This is really important to do, but our focus was on inducing cross protection. So do we have cross protection? Do we have T-cell response in the lung providing cross protection? The hypothesis and the whole strategy we work on, this will act like a live virus. It's supposed to induce interferon type 1 response, so you'll have a limited response. You may develop some fever, but because this is acting like a live virus, will induce this proper immune response. It will be very limited, and when we go into clinic, then we have to test to see how far we go with whether it will be applicable to children or no. This is has to be part down the clinical trials.
Has anybody ever looked at gamma radiated vaccines from a regulatory perspective? I'm just following on from midlife in terms of making it, what are the criteria to make sure that you don't have other things that aren't irradiated and what the effect of that?
Okay, I just showed two vaccines which is currently being tested clinically. The gamma-radiated melanoma vaccine, and the other one is gamma-radiated BCG. In terms of regulation, the regulator wants sterility testing. So we could talk about, as I said, I'm not going to talk about the nuclear physics aspect of it, because before we were so over-occupied with the nuclear physics aspect of gamma radiation. What the regulators want is sterility testing. So current vaccine tested in embryonic eggs. You get 2 mils inoculated into 10 embryonic eggs, 200 microliter per egg, and then 3 passages. If that is negative, that is negative. So in terms of gamma radiation per se is not an issue with regulators. They're only concerned about sterility of those products.
Do you have to get antibody responses also with this?
The vaccine will induce good antibody responses and will also seroconversion. And we measure antibody responses. Antibody responses will be crucial for homotypic protection. And so it will act like a seasonal vaccine. If you need a universal or cross-protective, then you need the CD8 T-cell response, and that's what's done by this vaccine.
Okay, final question, Jerome.
We haven't tested that one, but it's only for one virus that could cause this WSN, but this one is not supposed to really cause, even live one, not supposed to cause the blood-brain barrier. So we have not tested that one.
Okay, enjoy the